Hello everyone. Welcome to the Tiger in STEM webinar series in chemistry. I'm Neha Mehta from Queen's University, Belfast, and it is with great pleasure that I'll introduce today's chair for the session, Laia Delgado Calico. And Laia studied chemistry at the University of Barcelona, and then she moved to London for a research stay at Imperial College London, and then did a master's in molecular biophysics at King's College London. And she's about to finish her PhD in the physics department at King's College London, and she has been working on metallic nanoparticles with computational methods. She's also a visiting researcher at KU Leuven, and so we have a great chair with us and great speakers. And I would like to say that this is our last session, so enjoy it a lot and have all the thrilling and exciting questions to share with all of us. And welcome to Laia.
Hello, welcome. Thanks, Neha. I am Laia Delgado Calico, and I am very happy to be here today. We should start by noting the code of conduct for this event. It is available on the Tiger's website through the event pages, and one of the hosts will post a link in the chat for you now. Please do take a look at it. By attending this webinar, you're agreeing to abide by this code of conduct. There are two ways of communicating with us during this webinar. The first is the chat box. You can use this to chat to us as hosts or to everybody here. You can change the settings in the drop-down menu just above the typing area. Please try to keep the chat on topic during the talks. Please don't share any personal details, and above all, be respectful of everyone here. If you're having any problems, the chat is the best way to reach the team running today's webinar. Secondly, we will have time for questions after each talk. If you have a question for the speaker, please type it in the Q&A box so that we can pick it up at the end of the talk without it getting lost in the chat. We encourage you to write your question at any time. There is no need to wait until the end of the presentation. We also have live captioning for all the talks today. If you would like to turn the captions on or off at any point, you can do so from the live transcription button at the bottom of your screen. Sometimes it shows up with a little CC button. We are grateful to our transcriber One-to-One Captions for captioning these talks for us today. We will be recording this webinar today, and it should be available in the next day or two on the Tiger's YouTube channel. Finally, this is a free event. If you're in a position to do so, we invite you to make a donation to the Cowrie Scholarship Foundation, which wants to fund 100 disadvantaged Black British students through UK universities in the next decade. Please quote Tigers in the comment section of your donation. Okay, so let's start with our first speaker today. Is Haldrian Iriawan. Haldrian is a fourth-year materials student at Imperial College London. He was also an exchange student at the Department of Materials Science and Engineering at Massachusetts Institute of Technology. His talk is about the fundamental limitation of electrocatalytic nitrogen oxidation to enable green nitrate synthesis. Perfect, we can see your screen. Yeah, there you go.
Great, thanks Laia for the introduction. And hi everyone. I'm really excited to present my undergraduate thesis work on electrochemical nitrogen oxidation to enable green nitrate synthesis. So the motivation behind this work is essentially the many disadvantages in the way we make fertilizers today. Today's process involves multi-step processes. Typically, we start with the reforming of methane in order to make hydrogen gas, which is needed along with elevated temperature and pressure conditions to cleavage the nitrogen bond in the Haber-Bosch process to make ammonia. Ammonia is then fed into the Oswald process to make nitrates. And this fertilizer making process, in particular ammonia making, is very energy and carbon intensive, emitting 1.4% of global CO2 emission annually, or around half the global aviation industry. Not only that, the centralized facilities means these fertilizers need to be transported to agricultural fields. Inside of this complex multi-step processes, we can envision an electrochemical process where we can think about an electrochemical device powered by renewable electricity, taking nitrogen from air and water to make nitrates, which can be directly transported into the irrigation systems to fertilize our plants. To make this technology viable, we need to make this reaction happen fast enough, and therefore the catalyst is key. Some proof of concept studies have been reported in literature reporting electrocatalysts to oxidize nitrogen. However, as we have plotted here, these studies report quite trace amounts of nitrate, the majority of them being around 10 parts per million or even much less than that. This essentially points to the inherent challenge of this reaction. Intuitively, this is because this nitrogen bond is really, really strong. However, this trace amounts of the product being detected is also quite concerning, because we know that nitrogen contaminants from laboratory settings, which can come from the glassware, gas impurities, catalyst preparation, electrolytes, the membranes, and so on, can easily dwarf the measured products, leading to false positives. So without further sort of verification, it is quite difficult to say if these reported catalysts would work at all. In fact, in our recent review paper, we actually investigated the system mass and the production mass on the systems and find why nitrogen catalysis has experienced reproducibility issues. So in terms of nitrogen oxidation, current progress has been fueled by sort of trial and error catalyst testing, lacking some theoretical insights. In this case, density functional theory, which allows us to probe the energetics of the elementary reaction intermediates, can really help us pinpoint which steps are challenging and what is the bottleneck. So my master's thesis is basically all about answering the following questions. Why do experiments report trace amounts of nitrates? And is there a fundamental bottleneck to the reaction? So here are some of the results that I've collected. And so to start with, I've shown here the energy profile of an ideal catalyst. So here, the global reaction is a 10 electron transfer starting from nitrogen, our first step, into the product, which is our nitrates. The star denotes our catalyst, and then the starred species denote reaction intermediates adsorbed on our surface. I've modeled here, 10 steps of proton electron transfers, which correspond to the global reaction. And here at the equilibrium potential for our ideal catalyst, all the reaction intermediates are flat at 0, and allowing our reactant and the product to be reversible with each other without any uphill steps in between. However, this is an hypothetical catalyst, and real catalyst is not the case. So for a real catalyst like titanium dioxide on rutile 110 facet, we see that many of the reaction intermediates are actually quite destabilized. So, and in this case, the first few steps, the N2OH and N2OH, are very, very destabilized, and it requires such an uphill energy to form these species. And from computation, we find that because this is an electrochemical reaction involving an electron transfer, we can actually apply some bias in order to bring this down. And this step corresponds to around 3.55 volts SHE, or a bias of 2 volts. So this sort of inert surface of TiO2 can be contrasted with a more reactive surface, such as iridium oxide. So for this iridium oxide, in contrast, the first few steps, such as the formation of N2OH, and N2OH, and so on and so forth, are significantly less challenging. However, the most challenging step is actually the later steps, in particular, the final step, which is when we're trying to form our product. And the computed potential is around 3 volts, or a bias of 1.8 volts. So across these materials, highly oxidizing conditions are really required to drive nitrogen oxidation. So we can actually take a step back from these specific surfaces and look at general trend. So I've plotted here the free energy of the reaction intermediates here, which represent the stability of these species on our surface as a function of the N2 binding on the surface for the different types of rutiles computed. So here, what is really the fundamental limitation here is that the stability of these reaction intermediates are strongly correlated with each other in a linear fashion, and the gradients are quite similar to each other. either m equals 2 or m equals 1. And this linear scaling is very, very fundamental. To understand the implication, we can actually think back our rutile, titanium dioxide. Essentially for this, the dream is to mimic the profile of the ideal catalyst. What it means is we want to stabilize some of the reaction intermediates, N2OH and N2OH by quite a lot, and some other intermediates only by a little. But this selective stabilization of reaction intermediates is impossible because of the linear scaling here on the left. So in fact, if we look at the different materials, stabilizing the first few intermediates leads to overstabilization of the later steps. What this means is that it is impossible, our free energy profile will always be corrugated, and it is impossible to achieve the profile of an ideal catalyst. We can actually understand the limitation in a slightly different way. So here I plotted the limiting potential, which is essentially the potential that we need to overcome the reaction steps. And as we go down, it becomes more oxidizing. And here we actually see sort of like a volcano trend. On the right side of this volcano for these materials, these reaction steps are limited by reaction activation, where the first step is most challenging. However, in contrast, on the left side, the green step, the first step is not as challenging, but the most bottleneck step is the product formation. And the optimal catalyst is the intersection of the two, where the two steps are least challenging. However, we see that even at the optimal catalyst, the overpotential or the bias required is still very, very large, around 1.4 volts. We can actually contextualize this by looking at the competing reaction, because here in our system, we have a lot of water, and this water can be oxidized to form oxygen. So in the competing reaction, we can perform a similar analysis. And we see that at the optimal catalyst, the overpotential is 0.4, which is much smaller than the 1.4 volts in nitrogen oxidation. We can actually quantify this challenge by thinking that for our reaction, the barrier corresponds to our sort of this computed bias. And then at room temperature, 1 volts difference actually corresponds to 20 orders of magnitude in weight. So here we can understand that the reason why we're not really detecting any nitrates in our experiments is because at the relevant potentials where nitrogen oxidation can happen, oxygen evolution, the competing reaction, would be happening at the 2 dominant weight. So in conclusion, we have unveiled for the first time this linear scaling or a trans-based analysis, unveiling the suboptimal scaling of reaction intermediates, leading to an unfavorable volcano. And this results in oxygen evolution being much more favored over nitrogen oxidation. Future work would involve considering materials that can activate other reaction mechanisms, or we can actually think about using non-aqueous systems to minimize the activity of water to overcome the selectivity challenge. So finally, I want to thank my supervisors, Ifan, Professor Young, and Professor Olivia, for making this work possible, and a lot of people who have been really helpful in my learning journey in the past year. Thank you.
Great, thank you. Very interesting talk. Let's look now at some questions. So I have a question here from Matthew. Hi, Haldrian, do you think that transcription metals or transition metals or composites are more effective catalysts in terms of the efficiencies or energy usage?
Hi, thanks for the question. So at the moment, obviously due to constraints, we've only focused on rutile systems. This is kind of like a model system, although this has already been tested by sort of other experimentally. I would say that this is the starting point, unveiling favorable scaling and moving into alternative reaction mechanisms, alternative material systems can probably help circumvent this linear scaling. However, based on sort of other experiences in other reactions like nitrogen reduction and so on and so forth, typically even moving to like transition metals or composites this linear scaling is usually quite difficult to break. And so I think maybe a more promising research direction would be thinking about a material system that can probably activate an alternative reaction mechanism so that we can avoid the constraints of this direct 10 electron transfers.
Thank you. Yeah, and when you mentioned in the conclusions, like for future work, other alternative non-aqueous solutions, do you have any idea of what else could you use?
Oh yeah, so for example, the alternative reaction mechanism. So in here, in our all the steps that we consider here, to make nitrates from nitrogen, we actually get oxygen from water, essentially. Alternative reaction mechanism, for example, we can think about a material system that has very active surface oxygen on the surface, such that our nitrogen accepts nitrogen from our catalyst surface. So maybe we can explore some very reactive oxides like perovskites or spinel oxides and so on and so forth. And maybe for non-aqueous electrolytes, we can probably think about using some organic electrolytes to suppress the activity of water. However, this might be a bit challenging because it really depends on the stability window of these non-aqueous electrolytes. And we can see that nitrogen oxidation requires highly oxidizing potential. So the second alternative might be more challenging, but there are some select stable non-aqueous electrolytes that can be stable in oxidizing conditions.
Last question, another one from Matthew. Do you think oxygen formed during the byproduct can be recycled to prevent wastage or sequestered by future reactions, for future reactions?
Yeah, that is a great question, because like the way I frame this is that this oxygen evolution is kind of a bad thing, right? However, sort of oxygen evolution is oftentimes a desired reaction in water splitting cells. So there is definitely a potential to sort of combine this nitrate making device with a water splitting device to say make hydrogen. So maybe we can combine this electrolyzer for nitrate making with an energy storage device through hydrogen making. So that is also a possibility.
Okay, excellent. Thanks a lot for your talk and for your answers.
Great, thank you.
Now, our second speaker today is Eleanor Tether. Eleanor recently completed her final year as a natural sciences student, majoring in chemistry at the University of Birmingham. Her talk is about a little introduction to iron-nictide superconductors. You're muted.
Sorry, I thought I'd clicked it. Okay, let's try that again. Hi, everyone. So this is my talk, a little introduction to iron pnictide superconductors, which is based off the research I did for my undergraduate literature project this year, which was on pnictide superconductors. So my plan for today is I'm going to go through a little background knowledge on what a pnictide superconductor actually is. And then I'm going to talk in more detail about the phase diagrams of the iron arsenides, because this was my favorite bit of the project. So, here's my, okay. So first question, what is a superconductor? Superconductors are materials which, when cooled below a certain temperature, known as their critical temperature or TC, enter a superconducting ground state. And in this state, they can conduct electricity without any resistance, and they're perfectly diamagnetic. And it's these two properties that make superconductors currently and potentially even more very useful for a wide range of applications. But the problem is all the ones that we have at the moment need to be kept really cold because they have low critical temperatures. So there's a lot of research going into seeing if we can find a superconductor that works at room temperature and pressure. So if you're going to take one thing away from this slide, it's that we're looking for superconductors with higher critical temperatures, because that is what my project was focused on. So next question, what is a pnictide? The pnictogens are just the elements on the nitrogen in the periodic table. So pnictide superconductors are a family of superconductors, all of which contain at least one of these elements, normally bonded to a transition metal. And the ones I was looking at were the 1111 structure type, which look like this. And they all consist of a layer of a rare earth element tetrahedrally coordinated to oxygen and a pnictogen tetrahedrally coordinated to a transition metal. And I was investigating how their composition and their doping changes their critical temperature. Now, for the sake of this presentation, I'm going to be specifically talking about those where the transition metal is iron and the pnictogen is arsenic. So when the field of pnictide superconductors was first starting to emerge, others with the 1111 structure type had been found to be superconducting, but only with critical temperatures like 5 or 7 Kelvin, which is almost useless. And the iron arsenides have been found to be antiferromagnetic, not superconductors at all. But then in 2008, it was discovered that if you dope the rare earth oxygen layer with fluorine, superconductivity will suddenly emerge for these materials. So this is the temperature versus resistivity graph for one of these materials. And if you look at the thick black line, that is showing you the undoped compound. And you can see that they've marked a hump with T anon, which represents the point where it changes into an antiferromagnet. But obviously after this point, the resistivity is going up, so it's not a superconductor. But as soon as you start doping with fluorine, so looking at any of the other lines, you get the classic drop off to zero resistivity associated with superconductivity. Now, this information can also be displayed as a phase diagram. So we have doping level along the x-axis and the temperature along the y-axis. And you can see that as you start to dope the material, first of all, the temperature at which it switches from a normal material to an antiferromagnet, so that's the black triangles, that starts to decrease. And then suddenly the magnetism disappears and you have superconductivity instead. Now, this material was found to have a critical temperature of 30 Kelvin, which is not super high, but it's a lot better than what we were previously dealing with pnictide superconductors. And it's high enough for it to be classed as a high temperature superconductor. So this generated a lot of interest, and there was a sudden flurry of research into this area. And other superconductors were found, but one of the first things that was noticed is that the phase diagrams for these different superconductors were looking remarkably different based on how similar the materials are. So here are 3 of them. And for all of these materials, the structure is the same and the composition is almost exactly the same. It's just the rare earth element that differs. But you can see just by looking at them, the phase diagrams look completely different. And this alone is quite odd. But then the samarium compound, so the bottom one, it was found that the magnetism and the superconductivity were able to coexist. And that's represented by the shaded in area. And this is particularly odd because magnetism in these materials, as I mentioned, is antiferromagnetism, which normally spreads out across the whole of a material. Superconductors are diamagnetic, and you can't have a material be both antiferromagnetic and diamagnetic at the same time. So this seems particularly odd and possibly shouldn't be possible. So when I was investigating this area, I came across this, and I was very intrigued by first the fact that the phase diagrams look so different, and second by this magnetic superconductive crossover. So I decided to have a bit more of a look into it. And first I looked at several papers that were looking at the samarium compound. And several of them did also find crossover backing up the findings of this one, but others weren't finding any crossover at all, and no crossover was being found for the other materials. So nobody really knew what was going on. And then I found a paper on the cerium material, which also found crossover. So this is their phase diagram. And this is important because it shows that the crossover isn't something that's intrinsic to the samarium compound, it's coming from the structure itself. But moreover, the authors of this paper noticed that those papers that hadn't found crossover previously were using a technique called neutron diffraction to look at the magnetism. And those that had found crossover were using a different technique called muon spin rotation. And the difference between these 2 techniques is that muon spin rotation can detect magnetism at much shorter ranges than neutron diffraction can. So they came up with this idea that what's happening in these materials is as you dope them with fluorine, the magnetism is getting confined to shorter and shorter regions within the material. And at a certain point, it's confined to such small regions that first of all, neutron diffraction can't detect it, but also superconductivity is able to emerge around it. And this is marked on the phase diagram here. So you can see LR indicating long range magnetism, SR indicating short range magnetism. And it's once it's in the short range region that superconductivity is appearing. They also found evidence for this in their muon spin rotation data. So going from a doping level of 0 to 0.06, you can see just by looking at them that the graph looks very different. And this is indicative of a switch from long to short range magnetic order. So after I'd found this study, I was really intrigued by this, and I started to have a look to see if I could find any further evidence to support that idea. And sure enough, the papers I've been looking at that hadn't found crossover were using neutron diffraction, and the ones that had found crossover were using muon spin rotation. I then also found some muon spin rotation studies on other compounds. So we have this neodymium material and the lanthanum material, both of which found crossover. So it seems very likely that this is a unified phase diagram amongst these materials. So that's all well and good. Why does any of this actually matter? The suppression of magnetism in these materials is clearly linked to their critical temperature because they only reach their maximum critical temperature once the magnetism is completely suppressed. But moreover, the highest critical temperatures amongst pnictide superconductors have been found for these materials. And it's been suggested that it's something about the magnetism that enables them to reach these much higher critical temperatures. And for that reason, it's important to understand how the magnetism and the superconductivity are interacting in these materials in order to understand how higher critical temperatures can and may be reached, which is, after all, what we're looking for. Do you have any questions?
Thank you. Excellent. I really like the design of your slides.
Thank you. I found it on the internet. There is an image credit in that. And yeah, it's a fun one.
So there's a question from Matthew again on the Q&A. Am I correct to assume that muon spin diffraction would have a greater degree of accuracy compared to an X-ray diffraction to detect differences in magnetism?
Okay, well, I'm not particularly well-versed in the techniques themselves because that wasn't what I was focusing on for my project. As far as I'm aware, X-ray diffraction isn't able to pick up magnetism, but neutron diffraction can. And yeah, muon spin diffraction is able to do it more specifically, yeah, with greater accuracy than neutron diffraction can.
There are no more questions at the moment. I can ask, like, do you think this can potentially be used like in these trains that go, like these superconductors, like levitating trains?
Well, I mean, hopefully superconductors in general will one day be able to do that because that would be amazing. I think that pnictide superconductors probably aren't going to get there. The highest critical temperature that's been found for these was, I think, like 58 Kelvin, and the best we've got going at the moment is 92 Kelvin, which is still not great, but is a whole lot better. And so it hasn't got higher than the temperature of liquid nitrogen, which is the point we need it for it to be actually useful for us at the moment. These superconductors, we might be able to find something, but the research in them has kind of dried up. So I think these ones are probably not going to be where we're finding something at the moment, sadly.
Yeah, we had someone asking about what's the current record for the critical temperature, but yeah, you've already answered that. And
I think it's 92 Kelvin. Google it to be certain, but I'm pretty sure that's what it is.
And the last question from Matthew, are there significant differences between perovskites or nictide superconductors, and which one is more effective for possible use in industry?
I mean, again, I don't know a huge amount about this specifically, but perovskites, I think, are doing better than pnictides at the moment. I mean, perovskite is a structure type, and the pnictides are based on which elements are included in them. And there aren't any pnictide superconductors that have the perovskite structure types. Perovskite ones, I believe, are copper-based, and they're all doing much better at the moment. It's a copper-based one that's 92 Kelvin, so they're more likely to be useful. But who knows, something weird might show up out of the blue someday.
Okay, thank you. Excellent. Yeah. Okay, so our last speaker today is Dr. Jerelle Joseph. The title of her talk is How Cells Form Compartments Without Walls. Dr. Jerelle Joseph was born and raised in the Commonwealth of Dominica. She holds an MPhil in chemistry and a BSc in chemistry and mathematics from the University of West Indies and from Cave Hill campus in Barbados. She earned a PhD in chemistry from the University of Cambridge as a Gates Cambridge Scholar. For her PhD work under the supervision of Professor David Wales, a Fellow of the Royal Society, Jerelle developed computational methods to study large-scale structural changes in proteins. Currently, Dr. Joseph is a research fellow in physical and chemical sciences at King's College, Cambridge, and works in the lab of Dr. Rosana Colle-Pardo. Her research encompasses developing multiscale approaches to study how cells organize their contents via phase separation. To date, Jerelle has published over 18 peer-reviewed articles and 1 book chapter in the field of computational chemistry and biophysics. Alongside her research, Jerelle is an avid advocate for Caribbean-based mentorship. In 2017, she founded Cari Scholar, an organization that connects Caribbean students to established professionals and academics in their field of study. So when you're ready.
Can you see? Good?
Excellent.
Perfect. Let me just organize myself. Good day, everyone. Thank you so much to the Tiger and STEMM team for organizing such an amazing series. Today, we will discuss a process used by cells to organize their contents, and I will describe how experiments and simulations can be combined to reveal important molecular details of this process. Now, when we consider intracellular organization, we often picture different regions of the cell forming tiny membrane-bound compartments like lysosomes, mitochondria, and the nucleus. However, there are numerous microenvironments inside cells that are not enclosed by membranes. And for a long time, scientists wondered, how do these membraneless organelles arise, and how are their structures maintained without physical membranes? Over the last decade, groundbreaking experiments have proposed the transformative paradigm of liquid-liquid phase separation to account for these membraneless organelles. That is to say, analogous to the separation of oil and water into distinct liquid phases, under certain conditions, macromolecules, in particular proteins in the cytoplasm and nucleoplasm, exhibit weak attractive interactions with each other that drive them to condense and undergo liquid-liquid phase separation, forming oil-like droplets inside cells, which we generally refer to as biomolecular condensates. Now, experimental studies have revealed that these compartments can flow, wet surfaces, drip off surfaces, all characteristics of liquids, strongly supporting the viewpoint that biomolecular condensates are indeed liquid drops and that they are formed by the process of liquid-liquid phase separation. Now, in general, the study of phase transitions involves either directly or indirectly constructing phase diagrams for the system in question. And in the context of liquid-liquid phase separation, phase diagrams describe conditions under which the two liquid phases will coexist. That is, for a particular protein, a protein-rich phase and a protein-poor phase. Now, determining the phase boundaries for biomolecular liquid-liquid phase separation and characterizing the ensuing biomolecular condensates are important for several reasons. For example, biomolecular condensates may serve as important reaction centers inside the cell. They may also play a role in genome organization, wherein certain parts of the genome may be compacted within phase-separated condensates, thereby regulating how our genetic material is accessed and ultimately expressed. Liquid-liquid phase separation may also lead to the formation of condensates that act as super enhancers that strongly regulate transcription. And importantly, phase separation has been implicated in the progression of neurodegenerative diseases, wherein phase-separated compartments may function as precursors to the formation of gel-like states or pathological aggregates. Hence, for these and many other reasons, it is very important for us to thoroughly investigate the formation and dissolution of biomolecular condensates via phase separation. Now, as mentioned earlier, proteins are key components of these liquid-like compartments. More specifically, multivalent proteins play a leading role in biomolecular condensates. When we say a protein is multivalent, we mean that this protein encodes multiple interaction sites. And these interaction sites may take the form of individual amino acid residues, short linear motifs, or entire protein domains, and are generally described by Paco and others as stickers. Now, when multiple complementary interaction sites or stickers occur within a single protein, these stickers allow the protein to to participate in homotypic interactions, wherein we have multiple copies of the same protein interacting with each other in a favorable manner. And under certain conditions, these interactions lead to the formation of well-connected networks that can sustain these phase-separated liquid-like compartments. Hence, this is why protein multivalency plays a critical role in phase separation. At a fundamental level, we therefore want to elucidate the precise molecular details of these interactions. That is commonly referred to as the molecular grammar of liquid-liquid phase separation, and determine how this language of biomolecular phase behavior transforms under different environmental conditions. Now, in terms of the molecular language of liquid-liquid phase separation, so far we know that protein condensation can be driven by both electrostatic and hydrophobic interactions, including charge-charge, cation-pi, dipole-dipole, pi-pi stacking interactions. We also know that these interactions can be strongly modulated in different environmental conditions, such as changes in salt, pH, and temperature. Moreover, recent experimental and theoretical work have predicted reentrant phase separation for proteins and polymers that undergo liquid-liquid phase separation via homotypic interactions. For example, it was predicted that certain proteins demix into two liquid phases at low temperatures. Then if the temperature is sufficiently high, these proteins form a single phase. However, upon further increase in temperature, the system can re-enter the phase-separated regime, hence the term reentrant, and similar behaviors have been predicted for changes in pH and pressure. Based on these results and predictions, we therefore questioned, can the monotonic variation in salt also lead to reentrant phase behavior in protein systems, particularly for those systems that are stabilized by homotypic interactions? To answer this question, our experimental collaborators first probed the phase behavior of fused in sarcoma protein, commonly known as FUS, in different salt regimes. And they found that at low salt concentrations, the protein forms phase-separated droplets, while at intermediate salt, it forms a well-mixed phase. Surprisingly, at high salt, FUS re-enters the phase-separated regime. Importantly, the liquid droplets that are formed in the high salt regime are fully reversible. That is, if we adjust the salt back to the intermediate range, the system again heals a well-mixed phase. And it is also important to note that these condensates are observed at the usual low protein concentration found in cells, as well as at physiological temperature and pH values. Now, how general is this behavior? In addition to FUS, they also observed salt-induced reentrant phase behavior for several other proteins. And interestingly, for all the systems, the high salt condensates had similar size distributions and shapes as their low salt counterparts. And so macroscopically, the condensates are more or less indistinguishable. And so these results raise two fundamental questions for us. What are the molecular interactions that lead to the formation of condensates in the high salt regime? And are these interactions the same as those that sustain condensates at low salt concentrations? Now, we know that significant reduction in protein solubility upon addition of salt, which is termed the salting out effect, has been attributed to hydrophobic effects. Hence, we hypothesize that enhanced hydrophobic interactions coupled with weakened ionic interactions may be the key drivers of these protein reentrant phase separation at high salt. To probe whether this was the case, on the experimental side, our collaborators selected compounds that are known to act as disruptors of electrostatic and weak hydrophobic interactions, and then tested the ability of these disruptors to dissociate preformed FUS condensates in different salt regimes. And what they found was interesting. So at low salt, both types of disruptors were able to dissolve the FUS condensates, while at high salt, only the hydrophobic disruptor was able to dissolve the condensates. Addition of the electrostatic disruptors did not show any observable effects, suggesting that at high salt, where electrostatic interactions are screened out, phase separation is primarily a hydrophobically driven process. In the experiments, we also tested the ability of high salt to now induce phase separation for proteins that do not undergo phase separation under physiological salt. And one such protein is PR25. This is a highly positively charged peptide, and this protein does not exhibit liquid-liquid phase separation at low salt because in that regime, its homotypic interactions are dominated by arginine-arginine repulsion. However, at high salt, it was found that PR25 can undergo liquid-liquid phase separation on its own. It was also found that whereas the hydrophobic disruptor was able to dissolve PR25 condensates, the electrostatic disruptors did not show any observable effects, again suggesting that liquid-liquid phase separation in the high salt regime may also be hydrophobically driven. So now, armed with these important pieces of information from the experimental work, we then set out to computationally investigate the molecular forces behind the salt-induced reentrant phase behavior of proteins. And here we combine two complementary levels of resolution. Specifically, at atomistic resolution, we investigated the relative contributions of electrostatic and hydrophobic interactions among pairs of amino acids and how that changes as a function of salt. And then at residue level, we probed whether these changes are in fact consistent with protein reentrant phase behavior. So in our atomistic simulations, we first selected pairs of amino acids that can establish the various types of molecular interactions that may be important for phase separation. And we then probed the interaction between these pairs of amino acids using a well-established technique known as umbrella sampling. So in umbrella sampling, independent molecular dynamic simulations are performed at different points along a reaction coordinate. And for each simulation, the distance between the interacting pairs of amino acids are restrained using a Bayesian harmonic potential. This essentially allows us to sample the interaction between the amino acid at a given distance or in a given window. For each window, we obtain a histogram of the visited configurations, which we can then combine to obtain a free energy profile for the interaction. And using this approach, we were able to probe how the interaction strength between pairs of amino acids varies with salt. What we found was that at short distances, the attractive interaction energies between oppositely charged amino acids decreased monotonically with salt, while interactions involving uncharged residues in general increased monotonically with salt. Importantly, we find that interactions between basic and aromatic residues, that is your cation pi interactions, exhibit a more complex behavior, which yields considerable interaction strengths at both low and high salt. More generally, and perhaps most interestingly, we found that the strongest interactions are those where two amino acids in the pair both have pi electrons in their side chains. And this result suggested that at ionic strengths, where charges are screened out, pi-driven hydrophobic interactions may actually be important for sustaining liquid-liquid phase separation. This dominant role of pi-pi interactions are even more evident in cases where both residues have the same charge but have pi electrons in their side chains, such as the arginine-arginine pair. This pair shows a more or less repulsive interaction at low salt and a weak attractive pi-pi interaction at high salt. Together, these observations strongly support our original hypothesis that at high salt, protein condensation is predominantly driven by hydrophobic contributions. And now we wanted to see whether we were able to, whether these changes in our atomistic simulations were consistent with this reentrant phase behavior. So for that, we use residue level coarse grain simulations, and the coarse grain model that we're using, each amino acid is represented by a single bead, and the energy of the system is computed as harmonic bond energy terms, an electrostatic term, and a hydrophobic term, which that takes into account short range pairwise interactions. And we then tune the electrostatic and hydrophobic parameters based on the trends captured in our atomistic simulations in order to mimic the different salt regimes. To investigate whether a protein phase separates within this framework, we use a technique known as direct coexistence simulations, and this essentially involves simulating both liquid phases in the same simulation box. So we begin at low salt and we set the coarse grain model parameters. We prepare the protein system, which essentially involves multiple copies of the same protein within the simulation box. We perform molecular dynamic simulations. Once these simulations have converged, we can determine the densities of the coexisting phases. And if we repeat at a different temperature, after repeating at several different temperatures, we ultimately are left with a phase diagram for the low salt case, which is depicted here. Once we have computed the phase diagram for the low salt case, we then select a temperature that is close to but below the critical temperature in the low salt regime, and then change the model parameters to that of medium salt and the high salt case. Again, perform molecular dynamic simulations and observe whether we obtain liquid-liquid phase separation or not, where we can tell whether we have liquid-liquid phase separation by the existence of well-defined interfaces in our simulation box. Consistent with our experiments, we find that at low salt, first undergoes liquid phase separation, but PR25 does not. While at intermediate salt, first condensates melted, and finally at high salt, first re-enters the phase separated regime, and PR25 now undergoes liquid-liquid phase separation. It is important to note that when we analyze the molecular organization and densities of the first condensates at low and high salt, these properties are similar in both regimes. Hence, the system does in fact re-enter a previously encountered phase separated state. Collectively, our experiments and simulations reveal that certain proteins do exhibit a salt-dependent reentrant phase behavior, and that different molecular forces stabilize condensates at low and high salt. Now, today I have talked a lot about the interactions made by proteins and how these interactions can influence their organization. I will now briefly highlight some of the key interactions that help shape my STEM journey. So to start off, I grew up on the island of Dominica, and I have this picture here that I like to show people, not to be confused with the Dominican Republic. And I then moved to Barbados, where I completed my undergraduate studies and a postgraduate degree. And then I later moved from Barbados to Cambridge for my PhD work. After my PhD work, I joined the group of Dr. Rizana Colapado as a postdoc, and presently I work closely with Dr. Colapado as a junior research fellow at King's College. Now, my STEM journey on this slide, while it appears here as being quite linear, has been defined by extreme lows and great highs, from losing both my parents as an undergraduate student and dealing with severe imposter syndrome as a Black woman in STEM, to my amazing experience as a Gates Cambridge Scholar, and then securing an independent research fellowship at King's College. Now, importantly, throughout these highs and lows, my STEM mentors have served as the key interactions that help shape my journey. For example, when I moved from Dominica to the University of the West Indies in Barbados, I was encouraged and supported by a family friend who had also studied chemistry there. My journey from Barbados to Cambridge was strongly supported by my then research supervisor, who had obtained his PhD at Cambridge years prior. And during my PhD, I received unwavering support from my PhD supervisor, And finally, as a postdoctoral researcher, I have been fortunate to receive invaluable mentorship from Dr. Colapado, another woman in STEM. So we often question, how do we support women in STEM? And while there are undoubtedly many systemic barriers that need to be broken to ensure that women in STEM receive an equal chance at success, one way we can start is by ensuring that girls and women have good mentors to encourage them, to guide them, and to support their journeys. And so today, if you don't remember anything from my talk, I encourage each one of you to pay it forward, whether it be reviewing a student's personal statement, helping someone prepare for an interview, or simply serving as a listening ear. We can each serve as powerful mentors for girls and women in STEM. And with that, I would like to thank my amazing colleagues and collaborators on the project that I shared with you today, in particular, the members of the Colapado Knowles, Albert C. Hyman, and St. George Hislop Labs. I also like to thank Jitan Mittal and Greg Dinan for their assistance in implementing the course-grain model, and Papu for useful discussions that greatly improve the quality of this project. And I would not be here without funding, and so I would like to express my thanks to the funding bodies for supporting our work. And I now welcome your questions. Thank you very much.
Thank you, very inspiring. Thank you for sharing your journey with us and your research. I first ask a question from the chat. You mentioned disruptors that you use to probe which forces play a role in aggregation. Can these disruptors also be exploited as a mechanism for therapies in diseases like Alzheimer's, which is linked to protein aggregation?
So this is a very good question. So the disruptors that were used in the study, they essentially can break up weak interactions. So if the interactions are sufficiently weak, they can disrupt them. And so the fact that the disruptors in our study were able to dissolve the condensates gives us a very strong impression that the condensates are indeed liquid-like. Now, if you have a state where, so one of the things is that these condensates can age over time and become more gel-like or even mature into aggregates. And so if you were to use a disruptor in that type of system, what you would see is that the condensate would persist, right? So it would not be able to dissolve the condensates, because in that sort of state of matter, you have much stronger bonds. And so these simple disruptors would not be able to break the bonds.
Thank you. We have another question from Matthew today. Does genetic mutations lead to differences in liquid-liquid phase separation?
Yes, so that is a brilliant question, and Matthew, thank you for all your wonderful questions this afternoon. Yes, so genetic modifications do lead to differences in liquid-liquid phase separation. As you can think about, you have a mutation, and so you might have a different type of expression of the protein, and so instead of having one particular amino acid present within the protein, it might be replaced by another, and that effect can be sort of like a domino effect. So let's say, for example, there are studies that show that if you mutate tyrosine residues for serine residues, certain proteins do not phase separate as readily, because tyrosines are known to form this very strong pi-pi interactions, whereas the serines do not. And so, yes, so genetic mutations, post-translational modifications, all these can shift the phase boundaries and change the way in which a system, or whether a system can phase separate or not. Very good question.
We have a follow-up question from Matthew. Do you think this research may be useful for patients suffering from kidney damage, where the kidneys may not be able to reabsorb protein?
Where the kidneys
may not be able to reabsorb protein?
Oh, that's an interesting question. So the kidneys cannot reabsorb proteins. Well, I would have to look into it further to know what is the exact reason why it doesn't reabsorb the protein. Like, is it an environmental issue? But I would guess that the inability could probably be due to the state that the protein is in. And if that is a factor there, then you can sort of use this work to determine what changes in environment or what drugs that you need to develop to move systems out of these phases or to prevent them from forming these aggregates that are difficult to absorb. But I don't really know much about the kidney aspect of it, so I would have to look into it further. But Matthew is giving me very good ideas. Thank you.
We have a question from Francesca. You showed miscibility gaps in liquid-liquid systems, and miscibility gaps are also seen in solid-solid systems. Solid-solid phase diagrams also give lots of other more complex forms to the phase diagram, like eutectics, peritectics. Is there anything analogous in the liquid-liquid regime driving complex liquid structures?
Yes, so you can have different, so you can have different sort of like arrangements of the condensates. So some of them might be multiphasic, where you might have additional organization within the condensate itself, where some proteins might prefer to position themselves more towards the interface, whereas others more towards the condensate core. And depending on the properties of those proteins, what you find is that the surface electrostatics of the condensate changes depending on how the proteins are arranged. And so that would lead to the condensate behaving different than others. So say for example, if you have a condensate that is highly charged at the surface, what you will find happening is that these systems might show greater resistance to fusing, which is a property of liquids, but you might have this sort of like spectrum of different arrangements and different properties based on the molecules that are involved and whether you have multiple components or not. And so the complexity can change, and that would undoubtedly change how the phase behavior of the system is. Yeah.
We have another question now from Rachel. When we use phase diagrams to understand liquid-solid or solid-solid equilibria in material science, we also have to think about the kinetics of nucleation of new phases, and foreign bodies can be really important as nucleation sites. Given all the different objects within a cell, are these objects important as nucleation sites and a relevant factor in how compartments form?
Yes, great question. So yes, so one of the key things in this sort of space of biomolecular phase behavior is that a lot of these condensates are formed by proteins and RNA, and a lot of these proteins that undergo liquid-liquid phase separation tend to have RNA binding domains. And so if you introduce RNA into the system, what happens is that RNA acts as sort of like a seed that helps to recruit the proteins and drive them into the phase separated regime. So you do have molecules that can act as seeds and molecules that can also suppress phase separation. For example, if you introduce a protein that, say, for example, has a very low valency, it cannot form as much connectivity within the condensate, and so it sort of destabilizes the condensate. So some molecules inside the cell will act as sort of the seeds and the sort of like scaffolds that recruits and drive phase separation, whereas others can act as suppressors. So yes, it's a good question.
Okay, and we have the last question from today, again from Matthew. Do you think that the research on phase separation can be applied to process of waste separation, since sometimes animal excrement which contain protein can be leaked into the sewer systems?
Yes, so there's a lot of work going on now to see how we can use the lessons that we've learned so far, and the field is very young. I should stress that. So the lessons that we have learned so far to make functional systems. So the ideas about, can we develop systems that selectively partition certain molecules, sequestering them and excluding others. So these ideas sort of new ideas in terms of polymer physics and phase separate and the behavior of phase transitions. These are not new ideas, but in terms of the context of biomolecular phase separation, this is now a stage where we're sort of like integrating stuff that we have learned from polymer physics and classical nucleation to understand this sort of biological systems. So the same sort of principles that we learn can be extended to other systems, but I should stress that a lot of these ideas are not new. The field itself is what is new, and how those rules are modulated within the cell and thinking about the cell in a different way. That is the new part of it. But the ideas of selective partitioning and stuff like that is something probably you would have learned when you did separating fluids in like high school or undergrad, right? When you have oil and water and you add something and one partitions into the oil and stays out of the water. So those ideas and are not particularly new, but thinking about them in the context of the cell, that is such a complex environment in itself, is what makes it very challenging. Yes.
Yes, indeed. So we don't have more questions. Thanks a lot for your talk and for answering all these excellent questions.
Thank you for all the excellent questions. I was just going to say that. Yes. Yeah. And thank you for having me.
Okay, so this is the end. We would be very grateful if you could take a couple of minutes to fill out our short evaluation form. One of the hosts will post a link in the chat any minute now for this. Your feedback will help us to run the best events we can and secure funding for more such events in the future. So please do fill it in if you can. This was the last seminar of our Tigers in STEM webinar series in chemistry, funded by the Royal Society of Chemistry. We hope you enjoyed the series. Thanks for turning in and take care.